Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture we are going to see another biochemical test, that is lipid hydrolysis test. So we already talked 24 biochemical test, okay, and these 24 biochemical test is present into my channel. If you want to see it, you can visit to my channel and watch all the biochemical test. Okay, there will be also a playlist of the biochemical test. Okay, I will also give the link in the description box, then you will check it. Now today in this video we are going to see that what is lipid hydrolysis test. So lipid hydrolysis test is also called lipase test. Now lipid, what is actually lipid? We also know about this. Lipid, as we better know, that it is the most important component, and it is actually the biological component for all the living organism, and it have high molecular weight, and due to this way it can possess a large amount of energy. Okay, now lipid are fats, waxy and oily biocompounds. Moreover, lipid also contain fats, oils, fat soluble vitamins, steroid, phospholipids and many other hydrophobic amphiphatic chemical compounds. Okay, as we talked that lipids contain fats and TG, means triglyceride, and these fats and TG are actually the source of energy that is found in the animal and plant. Okay, now what's going on when bacteria use it? So how the bacteria use it? Bacteria actually, they release an enzyme which we call it lipase enzyme, and due to this lipase enzyme they can cleave the ester bond between these compounds, means between the fats and triglyceride, and when the bond or break down, what will happen? Energy is released, that is the ATP, by the addition of water molecule. Okay, when the water molecule is added, ester bond is break down, then ATP energy is released, and due to this way the byproduct is, means we get glycerol and fatty acid respectively. And when we want to check it, either the glycerol fatty acid is produced, so we get unpleasant odor. So this is how the lipid hydrolysis test, and this is a little bit introduction to lipase test. Now the objective of this test is that we specially use this test to detect the ability of the bacteria. Okay, to check the ability of the bacteria, either it can produce the exoenzyme like lipase or not. And when it produces lipase enzyme, so then we also check to see that either it can hydrolyze the lipid compound or not. Okay, so this is the objective. The requirement of the test is that we need a tributylene agar medium, or we also need egg yolk agar between these two media. Okay, so we can use any one of the media. Now next we need autoclave, incubator and personal protective equipment. Personal protective equipment means goggles, gloves, overall. So these are the personal protective equipments. We need petri dishes, conical flasks and other general lab material, like we need water bath, we need wire loop, we need spirit lamp, we need LFH. So all these things we need while performing that test. Now the composition of the media, let's suppose if the media is not available, if we have all these ingredients, so we can also make the media. So we need peptone 5 gram, we need yeast extract 5 gram, agar 15 gram. So when we mix it in 1 liter, so we get tributylene agar. So we also need tributylene, that is the TG. Okay, and the final pH is 7.5 at room temperature at 25 degree or 27 degrees Celsius. So this is the composition. The principle of the test says that as we better know that in this test we need what we actually use, the TG tributylene. Now this TG tributylene is actually found in the animal and plant fats and oil. And why it is important? Because it can demonstrate or detect the hydrolytic activity of exolipase enzyme. That's why it is important. Next, that we need neutron agar as well as triglyceride tributylene. When we combine both these, so then we get the activity of hydrolytic enzyme, hydrolytic activity we get, as well as the nutrient agar is actually providing the nutrients for the bacterial growth. That's why it is important. Now what's going on, this tributylene is actually depressed into the agar, and due to this way we get the emulsion. And one thing more, that due to the emulsion, what we get the opaque medium that is actually observe exoenzymatic activity. Another point says that when we perform the inoculation and incubation, after that what we actually observe, the bacteria actually secreting the lipase enzyme, and it can degrade the lipase, and after that what will going on, lipolysis will occur, and we get the zone of lipolysis. Okay, and it will be observed on the bacterial growth, around the bacterial growth. And when we get the hydrolytic reaction, okay, what does it mean? It means that it is the positive reaction, and during this hydrolytic reaction what we get glycerol as well as fatty acid. So this is the principle of the test. The test procedure says that first of all we need tributylene agar base powder, or if it is not available, so the ingredient which I tell you, we need also this ingredient. So we will mix it into 1 liter. After that we will mix the 10 ml of tributylene triglyceride. Now we will mix all these things and we will heat it. During this heating the ingredient will be mixed well. Then we autoclave it at 121 degrees Celsius for 15 minutes. After the autoclaving we will let the media to cool at 50 degrees Celsius. Then we will pour the media into the petri dishes, into sterilized petri dishes. After that, when the media gets solidified, so then what will we do? We take the wire loop, we will sterilize it. After that we will take the bacterial colony and we'll streak it into this media. Okay, now after 24-hour incubation, 24-hour, 48-hour or 72-hour incubation, we have to check the clear zone of tributylene hydrolysis around the bacterial colony. This is how the test procedure. The result, this is the result, as you can see. Now this is the positive result. As you can see, there is a zone, okay, clear zone. This is the negative result, there is no zone. Okay, this is another plate, so we get the zone. This, as you can see, there's bacterial colony as well as the zone of, this is the zone of, okay, and this is another bacterial colony, but there is no clear zone. This is another plate. This is actually the growth of the Staph aureus, as you can see, and around this growth we get the zone. This is another growth, as you can see, and this is the also Staph or Clostridium or Pseudomonas, and it can also produce what? The zone. So this is the result, or positive and negative result, as you can see. Now let's explain that what type of the bacteria are lipase positive and lipase negative. So as we talk that the positive result should be clear zone around the inoculation area, and the bacteria is actually Staph aureus, Staphylococcus saprophytex, Clostridium botulinum, Bacillus subtilis, Pseudomonas aeruginosa. These are the bacteria that are showing positive result. The negative result means that there is no zone around the inoculation, and the bacteria is actually Clostridium perfringens, Clostridium difficile, E. coli, Klebsiella pneumonia, Klebsiella oxytoca, etc. These are the bacteria that are showing negative result. The quality control, if we want to explain the quality control of the bacteria, so in this case let's suppose if we want to check that either the media is well, or that the media, if we want to check the quality of the media, so we have to streak the Staphylococcus aureus, that is positive control, and Clostridium perfringens, that is negative control. If let's suppose both the bacteria are showing opposite result or showing no result, it means the media is not working. Okay, so these two bacteria are very important to check the quality of the media. Now the uses of the test, means why we perform this test. So this test is especially important to check the lipolytic activity of the bacteria in food and dairy product. Next point says that this test is also important to differentiate and identify the member of Corynebacterium, Clostridium, Bacillus and Moraxella. It is also important to identify the bacteria that secrete lipolyze lipase enzyme, including the member of Enterobacteriaceae like Fusobacterium, Propionibacterium, Pseudomonas, Mycoplasma, and Staphylococcus. So this is the uses of the test. The limitation of the test says that if we want to check, if we want to identify the bacteria, so it doesn't mean that we should perform only this test. So for the bacteria identification, we need additional biochemical test, as we already talked. Okay, now this test requires longer incubation because there is also anaerobic bacteria, and anaerobic bacteria need 3 days. After 3 days it can show the result. Now another most important thing is that fastidious bacteria, fastidious organism are not growing on this media, and thus it is very difficult to identify this type of the bacteria. And one thing more, that if we want to get big and clear and quick result and clear and big zone, so we have to take a heavy inoculum. So this is all about the lipase test or lipid hydrolysis test. I hope this video is helpful for you. Thank you so much for watching.